Hey everyone, today we'll be covering Chapter 12. The topic of Chapter 12 is heredity. In essence, how does heredity work? Heredity is the transmission of traits from one generation to the next. So you inherit certain traits, certain features from your parents, and they inherited those from their parents, right? Your grandparents. So before we really go into heredity much, I need to tell you a little bit about the background. Before the 1800s, we really didn't understand how heredity worked very well. We didn't know why you had certain features, why you had brown hair, certain people had blonde hair, why you have brown eyes, other people had blue eyes. We really didn't know how it worked. People thought that the blood had something to do with it, hence the term bloodline. People thought that there was this blending that happened where if you have this feature, it blended with your mate's features and then you had some kind of blend in your offspring. And now we know that none of those things are true. You don't pass on your traits through the blood. Traits do not blend. And the reason we understand more about heredity and the things that we do about heredity are early pioneers like T.A. Knight and Gregor Mendel, who I'm going to talk about in a minute. But it really started around the 1800s. So what Knight did was he actually looked at some pea plants, and this was his genius contribution. He looked at the pea plant Pisum sativa, and this plant is really easy to study. This plant, you can take peas, plant them, they grow into pea plants, and then those plants are flowering plants. Let me just show you real quick what these plants look like. Here you can see it on the right. You can see the pea plant. It's a flowering plant. Here's a purple flower. There's the pea pod, and the peas would form inside. I just want to give you an idea of what these pea plants look like. Okay, take a look here. So these pea plants come in a huge variety. You can get pea plants with purple flowers, white flowers, you could study white versus purple trait. Yellow peas versus green peas, that's another trait you could study. Round peas versus wrinkly peas, green pods versus yellow pods, inflated pods versus constricted pods, flower positioning in the middle versus the end, and tall plants versus short plants. So pea plants are great to study because you can get all this variety to study heredity. Another reason why pea plants are so good is this. This was a huge deal. Pea plants are flowering plants. Flowering plants have male bits called anther and female bits called carpel. The male bits or the anther form the pollen. The pollen contains the sperm. All you have to do is the sperm travels from the tip of these filaments, these yellow filaments called anther. All the pollen has to do is travel to this tip right here called the stigma. The stigma is the end of the style, and the pollen or sperm then travels down the style, and here's where all the eggs are. Here's an egg, here's an egg, here's an egg, here's an egg, here's an egg. So did you see what happened? The same flower bud, the same flower has sperm and eggs. The sperm are at the tip of the anther. The eggs are inside of this structure called the carpel, right? The female part of the flower. So flowering plants can self-fertilize. What does that mean? Sperm is produced here, travels to here, travels down, and fertilizes eggs. You don't need more than one plant to have offspring. Okay, it's not like humans. Humans, you need a male and a female to mate, and then you have offspring. With plants, you just need one plant. One plant has the sperm, the same plant has the eggs, and that plant can fertilize itself. And look at this. What happens when you fertilize those eggs? Those become the peas. So each pea in the pea pod, each pea is a fertilized egg. It's an embryo. It's an embryo. And if you were to plant that pea, it would grow to become its own flowering plant. Okay, so very interesting stuff there. Okay, so again, why are these plants so important? Why are these plants so nice for studying heredity? Well, you could allow them to self-fertilize and see what the offspring of itself looks like if you self-fertilize, or you could cross-fertilize. You could take pollen from this plant and insert it into the female carpel on this other plant. So this would be called cross-fertilize, where you take pollen from one plant, let's say a white flower plant. You take pollen from the white flower plant and you deposit it on the purple flower plant. These are two different plants. That's called cross-fertilization. Or like I said, you could just go ahead and take the pollen from this flower and allow it to pollinate itself. That would be called self-fertilization. And that is an amazing trait of these plants. Animals can't do this stuff. You know, you can't take a mouse and get more mice by having that mouse mate with itself. It doesn't work that way. But with plants, you can. And so you can do all these extra cool things with plants. So with that background, let's just get started on Knight's contribution, T.A. Knight, and what he did in 1823 to help us along with our understanding of heredity. Let's go back to that first slide. So what did he do? He crossed some pea plants, but he didn't just cross any plants. Look what it says here. He crossed 2 true breeding strains. What does that mean? Okay, I need to explain this. Let me set up for this, and I'm going to explain what a true breeder is. All right, I'm all set up. Let's talk about what a true breeding plant is and how Knight's contribution to setting up crosses between true breeders was such an amazing revelation. So take a look here. If you have a yellow pea plant, a pea plant that came from a yellow pea, all right? If you were to self-fertilize that plant, self-fertilize, remember what that means, take its own pollen to fertilize itself. What would you expect its offspring to look like? What kind of pea color would you expect? Well, you might expect yellow peas, because if the creature itself came from a yellow pea, why wouldn't it produce yellow pea offspring if it mates with itself? If it produces its own sperm and its own eggs and it has its own offspring, they should also be yellow, right? So you would expect 100% yellow offspring, yellow pea offspring. And that's what he saw with some plants. Here's the weird part. Some of his yellow plants, if you allowed them to self-fertilize, they only give you yellow peas. And that makes sense, 100% yellow. Okay? But here was the weird thing he noticed. Other yellow pea plants he did that on would give you some yellow, but some green. That's weird, right? Why would you have green peas if you're a yellow plant who's self-fertilized? Why would you show any green peas at all? But again, how did this work? Some of his yellow pea plants, if you allow them to self-fertilize, those would give yellow pea offspring. But some of his yellow pea plants, if you allowed them to self-fertilize, they'd give yellow and green peas. So that's weird. I'm not talking yellowish green or yellowy green. I'm talking some of their offspring would be yellow, some of their offspring would be green. Okay, and to make things even trickier and weirder, he had green pea plants, and when he self-fertilized those, they were always 100% green. Right? So he couldn't find a green pea plant that doesn't give you 100% green results when self-fertilized. So it seemed like only the yellow ones could he find ones that would give you only yellow or ones that would sometimes give you green. With his green pea plants, if you self-fertilize them, they always give you green. Okay, this was something he noticed before he even set up his experiments. This was his big contribution. So what does this tell you? This tells you that some yellow pea plants he called true breeders. True breeding. What does true breeding mean? True breeding simply means for whatever trait you're studying, let's say yellow pea color, if you self-fertilize that creature, it will be 100% like the parent yellow. So he would have called this one a true breeder, this plant a true breeder, but he would not have called this plant a true breeder. You know why? Because this plant, when you self-fertilize, you get green and yellow. Okay, I should say green or yellow peas. So that would be called a non-true breeder. What about this green one over here, this green plant? Remember, it didn't matter which green plant he looked at, they were always true breeders. Why? Because when you self-fertilize these, they always give you a green result. Okay, so do you know what his big finding was? He would take true breeder yellows and cross them with true breeder greens. That was his big deal. He said, if you're going to set up a heredity cross, you have to set it up between true breeders. You want a true breeder yellow, and you want to cross it with a true breeder green. That's how we want to set it up. Now we can look at the next generation. He called this the parent generation. This is the P generation, or P1 generation for parent. Then you got the filial generation, the first generation, right? The children. And you know what he saw? When you cross a true breeder yellow with a true breeder green, do you know what he saw? Something very strange. 100% of them were yellow. Did you see that? So 100% of the children between the cross fertilization of this yellow pea plant, the true breeder, and this true breeder green gave you yellow. So at this point, he was very confused. You know, what happened to the green information? Did it disappear? Was it left out? Did it get destroyed? You know, what's going on? What happened to the green information, right? Well, here's another interesting thing he did. Look what he did. He took these yellow offspring and he self-fertilized the yellow offspring. And he said, okay, what do I get? What do I get when I do that? When I self-fertilize these F1 offspring to see the F2 generation, the grandchildren, right? You know what he saw? Some yellow and some green. So green comes back. You see that? Green made a comeback. It's like, whoa, where did green come from? Well, he speculated, well, these green that green information must have been in this yellow offspring, right? And I'm telling you, this yellow wasn't some greeny yellow. It was this yellow grandkid was just as yellow as this parent, is just as yellow as that parent. This green was just as green as that green. There's no blending going on. But green came back. What does that tell you about this guy, the F1? It doesn't just have yellow information, does it? It also must have green information. Does that make sense? This creature here has yellow information and green information, because when you self-fertilize it, you get yellow peas and green peas. Where did the green information come from? That parent. So did you see how powerful this stuff was? This showed Knight and Mendel after him that each individual possesses two versions of the same, what we know now is called gene, two alleles for the same gene, two versions of the same trait. So if the trait is pea color, every individual has two bits of information for pea color. So I could have yellow pea color information inside, and I can have green pea color information inside. But one of those pieces of information can mask the other, right? Have you heard of dominant genes or recessive genes? Have you heard those terms? That's what we've discovered. This creature must have yellow information, and yellow is denoted with a capital letter because it's the dominant color, and it has green information. Do you know how we show green? Do we write G? No, we write a little y. You see that? Big Y represents yellow. Little y represents whatever the recessive gene is. In this case, it's green. So this creature, its genotype, genotype meaning what genes does it have? It has to have a yellow gene allele and a green allele. Allele means version of that gene. Yellow allele, green allele for the pea color gene. Does that make sense? And what would Knight or Mendel have called this plant? Is this a true breeder? When you self-fertilize and you get green and yellow, is that a true breeder? That's a non-true breeder. Does that make sense? So we've just discovered what non-true breeders are, didn't we? Non-true breeders are individuals that have two different alleles for one gene, two different pieces of information for that one trait. So for example, if I have brown eyes, okay, I have two pieces of information for eye color. I could have brown eye information and blue eye information inside, right? Does that make sense? But does that mean I have blue eyes? No, because if brown eyes is dominant to blue eyes, even though I have blue eye information, I have got blue eye and brown eye information, I'm going to have brown eyes. This creature has yellow and green information, but it's a yellow pea plant because yellow is dominant to the recessive green, okay? So, and that's what a non-true breeder is. It's when you have two different pieces of information for the same trait, yellow and green, for example. What if you have two of the same information? Then you're a true breeder, aren't you? So what's the genotype? What are the alleles for this parent, this true breeder? Yes, if you said big Y, you're correct. True breeder means you have two of the same allele. So if I'm a pea plant and my genes say yellow and yellow, I have two alleles for yellow, and they say the same thing, that's called a true breeder, okay? And what about this one? This parent would be little y, right? Green, green. This parent was yellow, yellow. This parent was green, green. That's why they're called true breeders. When they have offspring, this parent gives yellow information, this parent gives green. So this offspring must have been yellow and green, and there you go. That's how it works. So you see how powerful this research was with the humble pea plant, Pisum sativa. It allowed you to self-fertilize, cross-fertilize, and all of that helped you to discover things like dominant and recessive alleles, or dominant and recessive traits. It helped you to understand that every creature has two alleles for every one gene. I could have a yellow and a green, or a brown and a blue, and whatnot, okay? And it showed that, you know, you could set it up this way, and as long as you start with true breeders, you're gonna have consistent results. So you have to always set up these crosses with true breeders, okay? So this is Knight's contribution. In the next video, I'm going to show you Mendel's. In my next Chalk Talk here, I'm going to show you Mendel's contribution with this monohybrid cross.
All right, so to pick up where Knight left off, in comes Gregor Mendel. He was a scientist, and he started his career actually when he went to University of Vienna. He studied science under famous researchers, including the person responsible for the technology or the physics behind the Doppler radar, Christian Doppler. Also some botanists. He learned how to work with plants. He learned how to work with numbers and how to set up good experiments. After that, his passions led him toward the monastery. He became a monk, an Austrian monk who worked, as a priest of sorts. And his passion for science, however, never left him. So he would tinker with genetic crosses, much like Knight did before him. He would set up experiments with the pea plant in the Abbey Gardens. So it was really interesting stuff. So here he is, he's a monk, but he's doing genetics experiments in the garden on the side. He chose to study the pea plant because of several factors. One, that you could form a hybrid. So, for example, you could cross a yellow and a green pea plant. You could cross purple and white flowers. Okay, many varieties are available. Okay, they're really easy to grow. Not only are they easy to grow, but think about it. After you finish your experiment, you could just eat the results. And you could self-fertilize them. Remember, that was a big deal, that you could self-fertilize or cross-fertilize these plants. Okay. So how do you do the experiment? You remember the flower is capable of self-fertilization. The pollen from the anther travels to the tip of the carpel called the stigma. Those sperm travel down and fertilize all of these eggs. Those eggs become the individual embryos, which you plant, and each one of those becomes a new pea plant. Okay, and remember, if you don't want to allow self-fertilization, you would simply snip the anther off before the flower is mature. So, to pick up where Knight left off, Mendel studied all these variety of plants. And here's an example of one of his famous studies called the Monohybrid Cross. Okay, you can write here, this is a monohybrid cross. Mono means one, because he was looking at one trait. And that trait in this particular example is flower color. All right, so let me show you how that works.
All right, so here I'm going to show you how the monohybrid cross works with Mendel and his famous monohybrid cross experiment. So what Mendel did was pick up where Knight left off. He crossed a true breeder purple flower plant with a true breeder white flower plant, and he got the F1 generation. And what did he see? 100% purple offspring. So what does that tell you about purple, the color? Is that the dominant allele or the recessive allele? Allele, again, meaning version of the gene or trait. Well, purple must be the dominant, okay? So, and remember, what letter does the dominant get? It gets the letter of the trait, so P for purple, and it's a capital P because it's the dominant allele. And so white in this case would be a lowercase p. So that's the genotype. Big P, little p, purple, white is the genotype for this hybrid. This is the hybrid. What would the parents be then if they're true breeders? Well, this parent, if it's a true breeder, remember true breeder means two of the same allele. This parent must be big P, big P. This parent must be big P, little p. You see that? I'm sorry. I hope you caught me. This parent must be little p, because this parent is a true breeder for white, which means it has white alleles. This parent is a true breeder for purple because it says purple. So I want you to think about this. Why is this offspring big P, little p? Well, think about it. Individuals are diploid, right? We've studied meiosis, and you know that individuals are diploid. That means they have homologous chromosomes. And what I want you to know is that this P would be on one homolog, and this P would be on the other homolog, right? So for example, if this big P is on its dad's chromosome 1, this other P would be on its mom's chromosome 1, right? Does that make sense? So when this creature makes sperm, let's say a sperm, do both of these big P's get packaged into that sperm? No, because sperm are haploid, right? So can a sperm have two P's in it, two big P's? No, sperm can only have one of the big P's, right? One or the other. So can you have a sperm with anything other than a big P in it from this parent? No. The sperm always say big P. What about this parent? Can you have eggs from this parent with 2 P's? No, it's either little p or little p. Did you see that? So why is that important? Well, look, if the sperm from this parent can only have a purple information, big P, and the egg from the other parent can only have white information, little p, what's the only option for the offspring? One of these sperm has to fertilize one of these eggs, right? So is there any other option besides big P, little p for the offspring? Because the sperm brings the big P, the egg has the little p. No, the offspring have to be big P, little p, which means all the offspring are going to be purple because big P makes you purple, right? And all of the offspring, all of this F1 generation has to be big P, little p. It has to be a non-true breeder. This means non-true breeder, right? And the parents are true breeders because they have 2 of the same letter, big P, or little p, little p. That makes them true breeders. All right, so what do we do from here? Well, remember what Knight did? He took these offspring, the F1, and he self-fertilized them, right? He self-fertilized them to see what's happening. So let's try that. If I take a big P, little p parent and I cross it with itself, isn't that like big P, little p crossed with big P, little p? Yes, it is. Now you have to ask yourself, what kind of sperm or egg, what kind of gametes could this parent make? Let's say it's the dad, right? How many, what could be in the sperm? Well, let's say it could make a sperm with what information? Big P. Or it could make a sperm with little p. Does that make sense? What about this parent? You know, it's mating with itself, so it's both parents. What could be in the eggs? What could be in the eggs? A big P could be in the egg or a little p. You just can't have 2 of the same letter in the same gamete, right? So it can make sperm with either big P or little p. It can make eggs with either big P or little p. So now you just solve. Look, what would happen if this sperm met that egg? What would be the genotype of the offspring? Look, big P, big P. It would be a homozygote, right? Homozygous dominant, they call it, because it has 2 big P's. It's homozygous. It has the same allele, and it's dominant because it's the dominant color. What would happen if this sperm met the other egg? The other egg, isn't that equally likely to happen? Then you would have a big P and a little p, right? Look at this. So this would be a non-true breeder purple, right? What would happen if this sperm met that egg? Oh, it would be little p, big P. I still like to write it with the capital letter first. Big P, little p. And finally, what would happen if this sperm met that last egg? That's right, you would have little p, little p. This would be the true breeder recessive, right? Recessive, homozygous recessive, which means true breeder, lower p, white, white. That has to be true breeder. So, and that's exactly what we saw. That's exactly what Mendel saw, okay? He saw, well, first of all, let's talk about phenotype. Remember what phenotype means? Phenotype means what we see, okay? The phenotypic ratio. You know what Mendel did? He did this in such high numbers that he got ratios that were reproducible, because he did this hundreds and hundreds of times. And when you do it hundreds and hundreds of times, you know what he saw? He saw 3 purples to 1 white. So what Mendel would say is the phenotype ratio or phenotypic ratio of the monohybrid cross. You're always talking about the F2 generation when you're talking about these ratios. The phenotypic ratio of a monohybrid cross, Mendel would say it's 3 purples to 1 white, 3 dominants to 1 recessive. Okay, because we had 3 purples for every 1 white. But now, think about this, wrap your brain around this. What is the genotype, the genotypic ratio? The genotypic ratio. that doesn't mean what you see. That doesn't mean purple and white. That means what alleles is possessed by these creatures. Alleles means letters, right? This creature here on the left, by the way, look, to confirm, when you self-fertilize this, look what you get. Only purples. So this parent was big P, big P. So how many big P's were there? One. Big P, big P. What are these two? Remember these two? These are heterozygotes, which means big P, little p. Big P, little p, non-true breeders. Remember? Non-true breeders. Non-true breeders. And, you know, just to confirm, like I said, he self-fertilized these, and he saw white come back. I know it's hard to see there, but white comes back. So you had one big P, two big P, little p's, and how many little p's? One little p. So this is a monohybrid cross. This is Mendel's famous monohybrid cross experiment. Okay? What do I need you to know? Again, the phenotypic ratio of a monohybrid cross is 3:1, 3 dominants to 1 recessive. The genotypic ratio of a monohybrid cross is 1 to 2 to 1. 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. Okay? Remember, homozygous means 2 of the same allele. Heterozygous means 2 of different alleles. In Mendel's time, he would have called homozygous true breeder. So that's homozygous dominant. That's homozygous recessive. And heterozygous, Mendel would have called non-true breeder. That means you have one of each allele. You have a big P and a little p. Okay, and that's it for that. So I'm going to explain now why this is important and what Mendel learned from this experiment.
All right. Oh, looks like we have a guest here. Oh, come on, Tig. Say hi to your fans. There you go, Tig. All right, Tig's come to say hi. Hey, Tig. All right, so let me show you. Hey, it's me and Tig. Ah, that tickles. All right, so remember, I just showed you how the monohybrid cross works, right? But what conclusions can we draw from that? Remember that the phenotypic ratio is 3:1. So 3 purples to 1 white. The genotypic ratio is 1 to 2 to 1. 1 homozygous dominant, which means big P, 2 heterozygous, which means big P, little p, and 1 homozygous recessive, which means little p. So what conclusions could we draw? Well, first of all, there were no intermediate traits, so blending is not a thing. Remember, the blending hypothesis is out. Okay, for each pair, he's rubbing his face on the keyboard. It's funny. Oh, Tig. For each pair, one trait was dominant, so purple was dominant in that example, and the other was recessive. White was recessive. All right, for alternative traits examined, pairs of alternative traits were segregated among the progeny in a particular cross. So what does that mean? So for example, every creature has 2 versions of every trait. So purple and white, or purple, white, and those they separate into the progeny, into the sperm and the egg. Okay, and remember, there was a 3:1 ratio, of purple to white. Okay, nowadays Mendel would have called them traits, but nowadays we know them as genes. Genes dictate our traits, right? Genes dictate our traits. And what is an allele? You should know an allele is an alternative form of a gene. So for example, if the gene is flower color, then the allele would be purple or white. If the gene is eye color, then the alleles would be like brown and blue or green. Okay, Again, remember what I've been saying? Homozygous. Homozygous means two of the same allele. For example, big P, or little p. That's homozygous. What would have Mendel called that? What would Mendel call homozygous? Mendel would have called it true breeder, right? Mendel would call it true breeders. And heterozygous, what would Mendel call that? He would call it a non-true breeder. That's right, it's a non-true breeder. Okay, and what other conclusions could we draw here? Remember, alleles remain discrete. No blending is going on. And then there were also dominant alleles. Oh, Tig's had enough. He's gone, you guys. So what else did we learn here? There was a dominant allele, right? Purple was the dominant allele, which is expressed, and you see the phenotype. And then there's a recessive allele. This is hidden by the dominant allele. This would have been, for example, the white flower phenotype. Okay, so I've been saying phenotype a lot, but what does that mean? It's the physical appearance. So white flower, purple flower, blue eyes, brown eyes, those are phenotypes. So what does genotype mean? Genotype means the total set of alleles in an individual. For example, the genotype would be big P, or big P, or little p. Those are all genotypes, okay? So that's how it works. And what did the monohybrid cross teach us? This is very important for you to understand. The monohybrid cross taught us this, the principle of segregation, that two alleles for a gene segregate during gamete formation and are rejoined at random from each parent during fertilization. So, what does that mean? Again, you have two alleles for every gene inside of a diploid creature. Like you and me, we have brown, right? Brown eye, or brown eye, or blue eye. But those two alleles segregate into our progeny. So remember, we are diploid. That's why we have brown, or blue, brown. Okay, we are diploid. But our sperm or our eggs are haploid, which means it either gets this one or that one. So a sperm can either have a blue allele or a brown, or one brown or the other brown. You can't have both alleles in one sperm. So your progeny gets one or the other allele, is what we learned from the principle of segregation of alleles from Mendel. And that is part of the big part of the reason he's known as the father of modern genetics. Okay, next I'm going to explain a Punnett square. So Reginald Punnett comes along after Mendel's experiments, and he says, "Hey, I can easily predict the outcome of these crosses. As long as I know the genotypes of the parents, the mom and the dad, I can set up one of these crosses and predict the phenotypes and the genotypes of the offspring using these Punnett squares." So let me explain how these squares work.
All right, so here I'm going to explain how Punnett squares work. So Reginald Punnett came along after Mendel showed his seminal work with monohybrid crosses and dihybrid crosses, and what Reginald Punnett contributed was his famous Punnett square. It's just a simple way of using this technique to predict the offspring genotypic and phenotypic ratios based on our understanding of the parents' genotypes. So if you if you know the genotype of the parents, you can easily figure out what their children are going to look like, what their children's phenotype's going to be, and in what ratio, what the children's genotype's going to be, and in what ratio. And you just set up the square. So for example, let me actually give you a better example. Let me show you kind of how these squares work. Sometimes it's better just to show you. This parent here, this was a true breeder recessive, right? So remember, its genotype is little p, right? This parent was big P. So now what you need to do to set up a Punnett square is ask yourself, what kind of gametes can this parent make? And what kind of gametes can the other parent make? And it doesn't matter if it's sperm or egg, but let's just say this parent's making the sperm. It doesn't matter. It could be making the egg. This parent could put what in the sperm? So what could be inside of a sperm? What information? Well, obviously a big P. Can you have two big P's in the same sperm? Can you have two big P's in the same sperm? No, because remember from chapter 11, meiosis, we talked about meiosis. The sperm and the eggs are haploid. They're only going to get one allele because they only get one homolog. They don't get both homologs. So you can only have one P, one allele for flower color inside of a gamete. So you can either make sperm with a big P or sperm with a big P. You're kind of limited to sperm with purple information. What about this parent? It can make eggs with little p or eggs with little p. There's no other options, okay? So the first thing is to know the genotypes of the parents. The second thing is to figure out what kind of gametes this parent can make and what kind of gametes that parent can make. And then how do you do a Punnett square? Well, very simple. You just draw up your square. And this is going to be a 2 by 2 square, or it could even be a 1 by 1 square if you want to simplify it, but I'm just going to make it 2 by 2. It can make these two types of gametes, which both actually say the same thing as big P, or it can make, this parent can make little p gametes. So let's do a 2 by 2 square. We could do a 1 by 1 square. So again, the sperm could say what? You put the sperm or male information, male looks like this, the male information on one side, and that's big P or big P. The egg information goes on the other side of the square. That says little p or little p, and then you just solve the square. If this sperm met that egg, what would it be? Big P, little p. If this sperm met that egg, it would be big P, little p. And these represent the fertilized egg and the new creature, the new plant, right? This one would be big P, little p. This one would be big P, little p. So what does that tell you? This Punnett square predicts that every single offspring is going to be purple because it has a big P. And every single offspring, oh, well, first of all, look, yes, every single offspring is purple. And every single offspring is that's right, a non-true breeder. It's heterozygous, which means big P, little p. It has two different alleles. And that's the case here as well. Because look, when you self-fertilize this, white comes back. So not only are all of the F1 generation purple, but also they are heterozygotes. They are big P, little p. See that? So now let's do the Punnett square. That was the Punnett square for the parent cross, wasn't it? Now let's do the Punnett square for this cross here. This parent, remember, is big P, little p. And if you cross with yourself, you know what that's like? That's like saying big P, little p crosses with big P, little p, because you're crossing with yourself, aren't you? Self-fertilizing. So, what kind of, I don't know, what kind of sperm can this parent make? Well, it's the same parent, but the sperm could say big P, right? Or, that's right, little p. How about the egg? What could be in the egg? Big P or, that's right, little p. So how do I set up my square? Very straightforward. You see, actually, big P, egg, or little p, egg on this side, and what's over here, the sperm information's on the other side. Big P sperm, little p sperm. Now let's solve this square. If this big P sperm met this big P egg, the offspring would be big P, big P. That's this, remember? Big P, big P. If this big P sperm met that little p egg, the offspring would be big P, little p. That's this offspring here. If this sperm met that egg, it would still be big P, little p. I know you might be tempted to write the little p first, but I always like to write the big P first. And then what if this, oh, and that's this one here. And what if this sperm met that egg? Then little p sperm would meet little p egg. And so look, there you go, that's your one recessive. And why is, and see there's a 3 to 1 ratio of purple to white, and it's a 1 to 2 to 1 1 to 2 to 1 ratio of homozygous dominant true breeder, heterozygous non-true breeders, and homozygous recessive true breeder recessive white. Okay, that's how the Punnett square works. And the Punnett square makes these crosses easier to do. You could do them without the Punnett square, but Punnett square makes things easy. And the reason Punnett squares work is because everything is an equal chance of working out. You know, the chances of getting this sperm are the same as getting that sperm, and this egg are the same. Because everything's equally likely to happen, these squares work out.
All right, so next I'm going to explain the dihybrid cross and how Mendel thought to do this extended experiment here. The dihybrid cross examines two separate traits in a single cross. So what you're doing is essentially looking at two different things at once. What he did was he produced true breeding lines for two traits, and he wanted to see what happens when you cross them. So look here, in this case, look at the two traits he's crossing. He made a true breeder over here. One parent is a true breeder of round and yellow. Okay, two different traits, right? Shape of the pea and color of the pea, round and yellow, and he crossed it with green and wrinkled. Okay, green color and wrinkled shape. All right, and before I get to the board and explain this for you, let me just explain why he cared to do this. Why did he want to look at the dihybrid cross? Why did he expand his experiment to include two traits instead of the monohybrid cross with just one trait? Well, here's why. He wanted to know if traits are linked or not. Okay, if they are linked or they are independent. So, for example, you see this pea over here at the top left? It's both round and yellow. Does that mean that round and yellow are always linked, such that any offspring that's round must also be yellow? And this one here at the top right, does any offspring of these two have to be, green or wrinkled or round or yellow? Or could you possibly end up with maybe or possibly could you end up with a round green, for example? Could you get a round green? Or could you get a wrinkly yellow? Again, are the traits round and yellow linked? Are green and wrinkly linked? Or can you mix and match? Can you get some combination that you did not see before? All right, so that is the main point. Let's hop to the board and explain how the dihybrid cross works.
All right, so here is how the dihybrid cross works. Remember, a dihybrid cross was Mendel's attempt to look at two traits at the same time. A monohybrid cross, you're looking at one trait, for example, flower color or pea color, that's it. In a dihybrid cross, you're looking at two different traits at the same time. So what two traits are we looking at here? Take a look. These are the parents, the parent generation. So this is some kind of true breeder cross with another true breeder. This one is true breeder for two traits. The traits are yellow and round. So the actual traits are color of pea and shape of pea. This one is a true breeder for yellow and round peas. This one would be another parent on a different plant. This is a different plant who's a true breeder for green peas and wrinkly peas, okay, for shape and color. All right, so when you take the true breeder round yellow and the true breeder wrinkly green, what did Mendel see in the F1 generation? That's right, look at this. 100% of the time, yellow and round. So what is your dominant color? Your dominant color is yellow, so yellow gets a capital Y. What is your dominant shape? Round, so that shape gets a capital R. So what would be the lowercase y? What would that represent? The recessive color, which little y would be for green. And what would be the lowercase r? What would that represent? Wrinkly, right? Little r is for wrinkly, big R is for round, little y is for green, big Y is for yellow. Okay, so this offspring told us that yellow is dominant, round is dominant, and this offspring is the hybrid. This is the hybrid, so it's a non-true breeder for yellow, and it's a non-true breeder for shape, for round. Okay, which means the parents are what? If this is a hybrid, remember the parents had to be true breeders. You always start these experiments with true breeders. That's what Knight taught us. So what would the genotype be for this round yellow pea? That's right, it would be yellow, yellow, big Y, and round, 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 big R, big R. What about this pea over here? It's green, green, so little y, and wrinkly, wrinkly, little r. You see that? Now, let's set up a Punnett square. Let's do a Punnett square to confirm this result. Let's do a Punnett square for this first cross. How do you do a Punnett square? Remember, I know the genotype of this parent, good. I know the genotype of this parent, good. What's the second step? Figure out what kind of gametes this parent can make. Well, what could be in the gametes? In a sperm cell. What could be in a sperm cell? Yeah, big Y and big R. Can you have two Y's in the same sperm cell? No, sperm cells are haploid. They can only have one Y. Can you have two R's in the same sperm cell? Again, sperm cells are haploid. They can only have one R, right? They only have one allele for every trait. You can't have a sperm cell that's YY, because that would suggest that the sperm cell's diploid. The sperm cell is not diploid. The organism is diploid, but the sperm cell is haploid. Does that make sense? So it can only have one of each letter type. It can only have one Y and one R. Is there any other kind of sperm that could be made, or am I done? I think that's it, right? And what about the egg? What could be in the egg? What could be in the egg? Well, it looks like a little Y could be in the egg, and can you have another Y? Nope, because eggs are haploid. You can have a little R though. You can have a little R. So you can have a big R, big Y in this gamete from this parent. You could have a little R, little Y from this gamete from this parent. So how do you set up your square? Very straightforward. It's a 1x1 square where the egg says little y, little r, and the sperm says, big Y, big R. So what happens when this sperm fertilizes that egg? Now you have a big Y and a little y, a big R and a little r. So 100% of your offspring should be yellow, yep, and round. And 100% of your offspring should be big Y, big R, little r. So non-true breeder for both traits. You see that? So we just used the Punnett square to show how we went from P1 generation to the F1 generation. But we're not done. Remember, we care about the next generation. We want to know what happens next. So what do we have to do? You remember? That's right, we have to self-fertilize this. Okay. And before I go on, let me just tell you, before I go on and show you how to self-fertilize that and what happened, what were the results, I just want to tell you, you might be wondering, why did Mendel even do a dihybrid cross? Why did he even look at two traits at the same time, color and shape at the same time? Why are we even doing this? Well, the reason he was curious about this was because he wanted to know if traits are linked or unlinked, right? So for example, if this original parent was round and yellow, are round and yellow linked? So the offspring have to be round and yellow, right? Is green linked with wrinkly? Is green linked with wrinkly? That would call them linked traits. Or do those traits assort independently? Or do they assort independently? What does that mean? That means, can you have grandchildren that are round and green? You know, like a combination you didn't have before, like round and green. Is that possible? Because that would suggest that the traits are unlinked. Can you get wrinkly and yellow? Again, that would suggest that the traits are unlinked. So what's going to happen? You know, that's what he was trying to get at. And actually, we're going to see that in the next slide. We're getting there. Okay, so anyway, I digress. So now getting back to my main point, how do I show this mating with itself? This yellow pea mating with itself. That means that one parent's big Y, little y, the other, and big R, little r, and it crossed with itself. So you have another big Y, little y, and a big R, little r. So that's both parents. Now remember, that's step one. What's the genotype of this parent? What's the genotype of that parent? You're mating yourself, so you're, both genotypes are the same. Now remember step two, to the Punnett square. What kind of sperm can this parent make? Let's talk about it. One sperm can have a big Y and a big R, right? Big Y, big R could be in one sperm, can't it? What could another sperm have? A big Y with a small r. Isn't that equally possible? Next one. Am I getting too low here? I think I'm getting too low. Hold on, I'm sorry. Let me write it. So remember, the first sperm was big Y, big R. The second sperm was big Y, little r. That's possible. The third sperm is, how about this? Little y with a big R. Little y with a big R. Is that possible? Yes. And how about the last type? Little y with a little r. Isn't that possible as well? All four of these gamete types, sperm types, are possible. Remember, you can't have two Y's, you can't have two R's, but you always have a Y and a R. There's four different Y-R combinations you could make. And if you're also the mom, well then, those are the exact same types of eggs you can make. Big Y, big R egg, big Y, little r egg, little y, big R egg, I'm sorry, it's getting messy, and little y, little r egg. So how is your square going to look? Look, how's your square going to look? Your square is going to look like a 4 by 4, 16 total squares in this cross for the dihybrid cross, because you have 4 different types of sperm and the same 4 different types of egg. So then you just solve the square, right? Remember how you solve the square? If this sperm meets that egg, you have big Y, big Y, big R, right? So this is going to give you a yellow and round offspring. That's a true breeder for both traits, big Y, big Y, big R. If this sperm meets that egg, you're going to get a big Y, a big R with a little r. So it's going to be yellow and round, true breeder for yellow, non-true breeder for round, right? And so on and so forth. And I'm not going to solve this whole square because it's solved on the next slide. I'm going to show you. But what if this sperm met that egg? Just real quick, what if this sperm at the bottom left met this egg at the top right? That would be little y, little r. And what would that give you? A green wrinkly, doesn't it? So let's check our work. I'm not going to solve this whole square. You know why? Look at the next slide. It's solved. Look, this is what we've been talking about. Look at this slide. Here are your 4 gamete types. I don't know, remember this was from the sperm we talked about. These are the sperm types from that parent. For example, big R, big Y, big R, little R, big Y, little R, little y, and then these were the eggs. The same thing. And remember I said the first pairing gives you big R, big Y, big Y. Yep, good. And the one at the bottom right, do you remember the one at the bottom right? That's the one that we said was little r, little y. So that's the little wrinkly green. So take a close look at this. Did Mendel answer his question? You remember the original parents? The original parents were round and yellow and wrinkly and green. Did we get any combinations that weren't in the original parents? Yeah, look. Didn't we get a round green? That didn't exist before. Didn't we get a wrinkly yellow? That didn't exist before. So what does that tell you about the traits? Are they linked or unlinked? They are unlinked, and they assort independently of one another. The traits assort independently. So what did Mendel discover with his dihybrid cross? He discovered independent assortment, that traits assort independently of one another. Very powerful stuff. And now we know because of meiosis 1, we talked about meiosis 1, metaphase 1, when independent assortment happens. Remember the tetrads line up any which way, randomly? This explains that. Isn't that interesting? So anyway, I will ask you on the exam, what is the phenotype ratio here, the phenotypic ratio of a dihybrid cross? Well, what is it? How many round yellows did we get? Round being the dominant shape, yellow being the dominant color. How many dominants did we get? Round yellow. If you count them, there's 9. 9 round yellows out of 16. How many dominant recessives did we get? I mean dominant for shape, recessive for color. So round green. How many round greens did we get? Dominant for shape, recessive for color. 1, 2, 3, 3. How many recessive dominants did we get? Recessive for shape, dominant for color. Those are the wrinkly yellow ones. That's 3. And how many did we get that are recessive for both traits? Recessive for shape, wrinkly, and recessive for color, green. Only 1, right? So what's the phenotypic ratio of a dihybrid cross? The answer would be 9, 3, 3, 1. Okay? Now, before I let you go, I need to tell you, if you see a question such as, what is the genotypic ratio of a dihybrid cross? That may just be a trick question. You know why? Because no one really asks what's the genotypic ratio of a dihybrid cross. You know what that means? What that question is asking you is, how many big R, big R, big Y, big Ys do you have? 1. How many big R, big R, big Y, little y's do you have? 2. How many big R, big Y, big Ys do you have? 2. So do you see where this could get ridiculous? It would be some ridiculously long ratio. It would be 1 to 2 to 2 to 2 to 2 to, it would be some really long, ridiculous answer. So if you see something like that, just a hint for you, what's the genotypic ratio of a dihybrid cross? The answer is most likely none of the above, because no one cares. I'm going to ask you, what's the phenotypic ratio of a dihybrid cross? 9, 3, 3, 1. And remember, what was the genotypic ratio of a monohybrid cross? 1 to 2 to 1. And what was the phenotypic ratio of a monohybrid cross? 3 to 1. Okay? So hopefully you understand that.
All right, so at this point, it's nice to know what is the takeaway message of the dihybrid cross. What did Mendel learn from the dihybrid cross? So let's take a look. Remember what I said earlier? Mendel was trying to figure out if you could get combinations that aren't present in the parents, in the original parents. Like the original parents were round and yellow, wrinkly and green. And remember, he wanted to know if those traits are linked together. Now look at the F2 generation. We did see round and green. That was a combination not seen before. And we found wrinkly yellow, another combination not seen before. So what does that tell you? Are the traits linked? No, the traits are not linked. You had round yellow, wrinkly green like the original parents, but you also got new combinations. Round and green assorted independently. Wrinkly and yellow assorted independently. So Mendel was able to determine the principle of independent assortment, that in a dihybrid cross, the alleles of each gene assort independently. And do you remember what makes sense of this in meiosis? We talked about meiosis just recently in chapter 11. Well, do you remember during meiosis 1, metaphase 1 How did those tetrads line up at the center of the cell? Well, they lined up via independent assortment. Isn't that neat? So now, without even knowing how chromosomes work or how genes worked, Mendel was able to anticipate that. He was able to determine that using nothing more than the humble pea plant. Okay, so again, what did the monohybrid cross teach us? It taught us the principle of segregation of alleles. And what did the dihybrid cross just teach us? The principle of independent assortment, that different alleles of a gene assort independently. Awesome. Next, I'm going to explain how a test cross works. A test cross is a simple genetic test that you can do to figure out a genotype of an individual with a dominant phenotype. Okay, so let me take it away at the board.
All right, for the last concept on this chapter, we're going to talk about what is a test cross. A test cross is a very straightforward test that you can do to figure out the genotype of a creature with an unknown phenotype. I'm sorry. Start over. All right, so this is the last concept of the chapter. This is the concept of a test cross, which is a very simple cross you can do to figure out the genotype of a creature with a dominant phenotype. Okay, so what do I mean? Let's say I give you a purple flower plant, right? I just say, here's a purple flower pea plant. And you know that purple flowers is the dominant flower color. You know that. Do you know the genotype of that plant I just gave you? Again, I gave you a purple flower plant. All you know is that purple is the dominant allele. Well, do you know the genotype of that purple flower plant I just handed you? Do you know if it's big P, or big P, little p? Do you know for sure? You don't know. You don't know. That's when you do a test cross. A test cross is done to figure out the genotype when you don't know the genotype, but you know the phenotype. The phenotype is the dominant phenotype. Now, would you need to do a test cross if I gave you a white flower pea plant? Would you need to do a test cross? No, because it has to be little p, doesn't it? If I give you a white flower plant and you knew that white flower is the recessive flower or recessive color, then there's no need to do a test because you know, oh, well, it must be little p, right? But if I give you the dominant phenotype, purple, and I say, here's a purple flower plant, then you would need to do a test cross to figure out, is it big P, or big P, little p? And how do you do it? Very simple, very simple. All you do in a test cross is cross your mystery plant with a white or a recessive, right? With a recessive plant, right? So if it's big P, and you cross it with little p, you're going to get a certain set of results. If it was big P, and you cross it with little p, you're going to get different results, right? So you could set up your Punnett square. This Punnett square, the sperm could get big P, okay? And the eggs could get little p. So all of your offspring are going to be big P, little p. Are you going to get any white? No. 100% will be purple offspring. But if the plant I gave you was a big P, little p plant, what kind of sperm could it make? Big P sperm, right? Or little p sperm, right? So if I set up my Punnett square, what could be in the eggs? Little p could be in the eggs. So in this case, big P, little p would give you a purple flower. And if this sperm met that egg, it would be little p, 50% white flowers, 50% purple. So did you see what happened? I gave you a purple flower plant. You didn't know if it was big P, or big P, little p. What you had to do was cross it with a white flower, a recessive plant, and depending on the outcome, you were able to determine the genotype. If you got no white offspring, the plant I gave you was a true breeder. If you got half white, half purple offspring, the plant I gave you was a non-true breeder, big P, little p. Okay, hopefully that makes sense.